Hello, everyone. Welcome. I see there might be some microphones on, so if you could switch them off, thank you. We are still waiting for until it's 3 p.m. sharp, so just opening people that people are able to come in from the lobby. But we will wait until 3 p.m. to start the actual agenda. But welcome, everyone.
Tanto que participar nem tanto.
Okay, welcome everyone. I see there are still people coming in through the lobby, but it's 3 p.m. sharp, so I think we can start this today's webinar. So good morning, good afternoon, and good evening, wherever you are joining us from. Welcome. On behalf of the Wastewater Surveillance for Africa initiative, I'm delighted to welcome you to today's webinar. This is actually our second webinar in our Wastewater Surveillance for Africa webinar series, which aims to build knowledge, foster collaboration, and spotlight innovative practices in wastewater and environmental surveillance across the continent. Today's theme is particularly important as it underscores the central role of collaboration, which is a key function of WES that brings together all the stakeholders from many various sectors, including public health, environment, and water. So welcome all, and before we will begin, I will briefly go over a few housekeeping points, just that we are in the same page with everyone. So just a second, I will share. So just few points about the webinar. So this session is being recorded. I believe there might be some microphones open at the moment, so if you could kindly close them. So this session is being recorded as it will be used as an upcoming training program that we will have. We kindly ask you to stay muted during the presentations and use the chat for the questions. And there will be time for live questions after each session. And most of all, be respectful and inclusive throughout the whole event. And yes, so next, I would like to also thank my colleague Valid, as we are also having the French interpretation throughout the event, and you can choose this from your own team settings. We want to encourage everyone to take actively part in the webinar, as highlighted also through the housekeeping rules. So please feel free to share your thoughts and comments in the chat at any time. And you will also have the opportunity to ask questions during the discussion sections. And we are very happy to hear your voices. And that's also why we hope that you will all take a moment to answer the post-webinar survey at the end of today's session. And next, I will hand the floor to Julian Blanc from UNEP One Health, who will kindly share some welcoming remarks. So over to you, Julian.
Thank you very much, Taro, and it's my pleasure to welcome all of you to this important event highlighting effective One Health in practice. Using wastewater and environmental surveillance for health is exactly the kind of cross-sectoral collaboration that the One Health approach promotes. We all know that human health, animal health, and ecosystem health are all deeply interconnected. And we know that effectively addressing the challenges arising from this interconnection, from this nexus, requires collaboration between sectors. But traditionally, the environment sector has struggled to identify its niche in the One Health approach as it is currently practiced. One Health originally was spearheaded by the human and animal health sectors, and it has long held onto a focus on zoonotic disease and antimicrobial resistance. But if we change that framing and we look at things from an environmental lens, new opportunities for collaboration and forward thinking and solutions emerge. Now, wastewater surveillance demonstrates the value of a truly multi-sectoral One Health approach with a strong environmental dimension. By gathering information about the pollutants and the processes that threaten the environment, we can also inform policy to simultaneously protect ecosystem health, animal health, and human health. And the Wastewater for Surveillance for Africa initiative is operationalizing this. And it's great to see representatives from diverse agencies from within the wider UN system, as well as other entities coming together through this initiative and through this webinar today. So while this is a step in the right direction, we still have a lot more to do. We need to foster exchanges such as this one, which is a great, as I say, a great starting point and a great step. But we need to highlight good practice examples of One Health in action that broaden that scope that I was previously so restricted. We need to catalyze innovation and prioritize interventions where collaboration makes sense. And we need to make the inclusion of the environment sector more than just an add-on or more than a box to be ticked, but to become an integral component in relevant interventions of One Health, actively capitalizing on the sector's strengths and expertise. In order to do this, we need to change our perspectives and ensure that the health of the environment is better valued and understood as a prerequisite for our own health. And the One Health approach can be a powerful framework to do this if we use it right. So thank you to our colleagues for organizing this webinar and for all the speakers and participants. And I very much look forward to hearing more about the West and Wastewater Surveillance for Africa initiative as it moves into practical interdisciplinary implementation. Thank you.
Thank you. Thank you. Many thanks, Julian, for setting up the stage for the webinar so well with your opening remarks and highlighting the importance of the collaboration. Today we have 3 very interesting presentations with the great presenters. And next, I would like to introduce our first presenter. Jean-Pierre Nusabimana is a leading expert in genomics and bioinformatics at the Rwanda Biomedical Center and program lead for One Health at the Africa Center of Excellence for Sustainable Cooling and Cold Chain in Kigali. Jean-Pierre is going to present us about how to embed WES in One Health policies in Rwanda. So Jean-Pierre, the stage is yours.
Thank you. Thank you, Taru, for the introduction. So I'll spend around 20 minutes talking about how Rwanda is embedding wastewater and environmental surveillance in One Health policies as an approach to involve for a multi-sectoral approach towards to solve issues such as AMR and others that are threatening our health. So I will give you an overview of the One Health approach in Rwanda and the implementation of the One Health strategic plan and the areas of focus with a particular focus on West, and also talk about the future of this initiative. So actually, the One Health coordination started taking place from January 2011 in Rwanda, where this initial phase was focusing on mapping and engaging different stakeholders. And different documents have been put in place around 2014, where we had the first strategic plan developed, and also GEEs and NAPs and other documents that are important to drive the One Health agenda at a national level. And as you can see, AMR surveillance tool was introduced very recently. So here it's a highlight of documents that have been put in place as per the chronology that I've shared above. And so for better coordination of One Health, it's recommended that there is a systematic approach to ensure that all the sectors involved are well coordinated. So this is the map of the structure of One Health multi-sectoral coordination mechanism that was established in Rwanda, where Rwanda Biomedical Center plays a major role as the secretariat and the coordinator of all the technical working groups that have been put in place. And these technical working groups come from different sector of development, such as agriculture, environment, and health. And we've established at least 5 technical working groups, such as food, crops, plant, drug safety, AMR being one, disease control surveillance being another, environment and disease management, One Health workforce development, resource knowledge management, and then advocacy and community. So key priorities include improving antimicrobial stewardship for the management of antimicrobial resistance, strengthening the global health security, pandemic preparedness, strengthening the community and health system resilience, and then reducing the social, cultural, and economic impacts. So our strategy to ensure that this mechanism is working, so we are working on establishing an integrated surveillance system where we collect data from environment, from the wastewater, and also from the patients, and at some point link them with epidemiological data that has been routinely collected by many countries using DHIS2 supported systems, so that we are able to respond to diseases in a holistic manner. So as you can see, this is it's very complex. I don't know if everybody can see it, but we're trying to show how everybody in terms of institutions are involved in the data flow from the environment, the hospitals, the abattoirs, so that at least we make sure that everything is converging to one place for a comprehensive picture of what's going on in our communities. So in terms of testing, we are implementing multi-pathogen genomic sequencing as a way to ensure that we don't miss out any important threat in terms of outbreaks or other threats such as AMR. And as you can see, the strategy is you start with multi-pathogen detection and moving to a single pathogen once you get we are able to detect threat. So we've started optimizing this approach, starting from the aircraft. And here is a picture showing how we've started collecting samples from the aircraft that come at the international airport of Kigali. And once these samples are collected, they are processed and sent to the National Reference Laboratory to be examined and inform the public health action. So here we've been collecting both, we started by collecting both wastewater samples and also nasal swabs from air travelers who volunteer to give the samples. And here is I'm showing a snapshot of our dynamic dashboard that shows example of SARS-CoV-2, which we started to use to validate this tool. So it's clearly showing that there is still need to optimize the tools to better fit the complexity related to wastewater samples. So as you can see, there's still more need for optimization to be able to call for variants, for instance, or be able to be specific enough depending on what you are trying to look at. So yeah, I've shown just a few examples of what we've been able to achieve. So example showing SARS-CoV-2. As I highlighted, we've been able to detect the new variants, but so detection goes better in nasal swabs, but we still need to improve on how we detect that in wastewater samples. The same with other types of mutations, where you see that it takes really quite some time to get it in wastewater as compared to the clinical samples. So, but in terms of positivity, you found out that you can detect, you can have more positive samples in wastewater as compared to nasal swabs, because as you know, with individual clinical samples, you need really a very big sample size to ensure that it's representative as compared to wastewater samples, where you only need up to a minimum of one sample to represent the whole population. So in case represent the population that occupy the aircraft, the specific aircraft where you are sampling from. So other things that we're trying to do is, so once we've optimized the tools to ensure that we are able to detect what could be the variants that we would be expect in the near future, so that we enhance preparedness and be able to optimize response on time. Something else we observed is that with aircraft, the longer the flight, the higher the positivity. So here is one paper that summarizes the data that I have just presented. So after learning several lessons about based on aircraft wastewater, so we've thought to expand from the aircraft now to the community wastewater-based surveillance. And we have identified different sites, around 10 sites in the country, as you can see on this map. And we are in the middle of preparations to ensure that all these collection sites are onboarded. And then also we expand not only in terms of the geolocations where we collect samples, but also in terms of the pathogens that we are looking at. So here I'm showing a new collaboration that we've started with UNEP to establish what I've just described above. And our major methods that we want to develop are based on shotgun metagenomics. So this is a kind of tool that is unbiased as based on pathogens, which is able to detect all the pathogens with no bias to specific pathogens. So, and for us to be able to implement this, we need also to establish methods that are pathogen agnostic, that would help for the shotgun metagenomics to work. So part of what we are developing is comparing different sample collection tools to see which one work better in different settings, and also be able to compare different methods of concentration, because here you collect in a very big amount of sample and you need to concentrate in a small amount to carry in the laboratory processing. So you need to choose the right tools that would be able to give you enough of the materials of good quality that you are able to carry for further analysis. So these are the things that we are planning to do, and also continuing to combine wastewater with other environmental surveillance aspects. So yeah, I'm showing pictures of the surveillance that we are doing with the bats. So one particular area that we are also working at is something to do with antimicrobial resistance in wastewater. Can I still be heard?
Yes, we can hear you. Yes, go ahead.
Yeah. So because we feel that this has been overlooked, like MR in wastewater. So, and we've here we are showing that not only us, but also many countries have. There's so many studies in different countries that show that wastewater can be a good tool, a good sample where to deal with antimicrobial resistance. So our objective is to engage with different stakeholders to assess and improve monitoring of AMR and treatment options in wastewater, identify potential technologies that can be implemented in Rwanda. So together with other partners such as the World Intellectual Property organizations. So among the technologies that we are looking at include chromatography-based techniques, the immunology such as ELISA, sensor devices, and also lateral flow. And we are exploring looking at different approaches for wastewater treatment to fight antimicrobial resistance, such as membrane bioreactor system, advanced oxidative pressures, among many, many others. So I'll stop here, and I'll take also this opportunity to thank many collaborators and partners that have been working with us in this area. And I will welcome any question or any comments during the Q&A session. Thank you so much.
Thank you so much, Jean-Pierre, for that presentation. Please feel free to put in your questions in the chat. Once we finish the presentations, we will have a Q&A session. Now over to our next presenter, Dr. Bernard Ouma Mware, who is a molecular biologist and virologist specializing in genomic surveillance and wastewater-based epidemiology at International Livestock Research Institute. Welcome, Dr. Bernard. Dr. Bernard, we can see your screen. You can go ahead. Dr. Bernard, sure, you have unmuted yourself. Yeah, we can hear you now. Go ahead. Yes.
Yeah. So good afternoon, good morning, and good evening, everyone. I started by thanking Josephine and team for giving us a chance from ILRI to just present on our experience, particularly on how One Health approaches are being applied in wastewater environmental surveillance here in our project within Kenya. So the presentation will focus on just the principles around One Health, look at antimicrobial resistance and the burden on health, and as well look at the cross-sectoral surveillance of pathogens, and finish with the experience of our work here at ILRI. Our focus is on shotgun metagenomics for wastewater surveillance. So as already been indicated that One Health is a comprehensive strategy that really focuses on the interconnectedness between human, animal, and environment. And the focus of One Health is basically to ensure that whatever we do leads to healthy outcome for both the livestock, human being, as well as environment. It focuses on, as my colleague has presented, multi-sectoral, interdisciplinary, focusing around collaboration, coordination, and capacity building, as well as communication, to ensure that then we have a healthy ecosystem and healthy human and animals. At ILRI, One Health focuses on a number of themes, and two of them which are of interest for this particular presentation is antimicrobial resistance and the issue around zoonosis, which is a key mandate of ILRI in terms of addressing livestock diseases as well as epidemics that could be affect both human and animal. At ILRI, we have a dedicated antimicrobial resistant unit that focuses on both really the microbial culturing and all the other techniques of screening antibiotics for their response or effectiveness against specific pathogens, and also doing community surveillance. But our focus from the genomics platform is using shotgun metagenomics to pull out signals of antimicrobial resistance, but also support the tracking and monitoring of pathogens that could spread between human and animals using metagenomics approach. Of course, we have themes around food safety, environment, wildlife, and climate change. Looks like my slides are not moving. Hello?
We can hear you well, Bernard. Yeah. Is it possible to try to click the slide, or is it completely stuck? Oh, now it seems to move.
It is moving.
Okay.
Yeah.
Perfect,
So
thank you.
the interconnectedness that exists around One Health comes also with opportunity of movement of pathogens, movement of microbes around humans, animals, and environment. And that is a great challenge to humanity. The fact that you'd have possibility of microorganisms moving between these three systems is a challenge. And I think one of the focus at ILRI has been to support the tracking and monitoring of zoonotic pathogens that could move between these systems. Of course, of interest, as I indicated, is the issue of antimicrobial resistance, looking at antibiotics flow and circulation within communities, as well as within the livestock sector. In terms of the resistome, we are aware that within the human sector, there's a use of antibiotics. Even within the animal sector, use of antibiotics. And through the waste from human and the waste from animals, these resistome or antibiotic resistance genes could find their way into the ecosystem, and from there they could be picked back and forth between humans and animals. And that becomes a challenge in the context of One Health. How do we address this interconnectedness to ensure that the possibility of movement is tracked, understood, and strategy that could be able to mitigate against transfer of resistance put in place? And so really just an overview in terms of antimicrobial resistance, which is known as a natural process through which microorganisms, including bacteria, fungi, viruses, are no longer responsive to medication, and that is likely to lead to a risk of disease spread, severe illness, or even disability to some extent, including death. And there are a number of factors that could lead to this. One is the idea that human beings may not be able to, have sufficient prescriptive medicine in terms of the ability to afford. And so the practice around medication may lead to this, as I indicated also, the issue of use of antibiotics between animals for growth as growth hormones, those could lead to this. And antimicrobial resistance impacts, in different forms. And it has been estimated that, within the next, 3-4 decades, we are likely to lose more than 10 million individuals. And that, if you think of communities where, population is 200, so it is a catastrophic, situation that may lead to many deaths. In terms of even the younger children, it is estimated that 1 in 5 is related to antimicrobial resistance. So this is a global threat to humanity, and therefore opportunity to develop tools that can be able to support tracking, monitoring, and strategies to mitigate are useful. This is basically just to highlight specific examples. On the left, the y-axis, you can be able to see the different pathogens, and then on the x-axis, you can be able to see the specific antibiotics and the statistics in terms of number of thousands of people that have, been diagnosed or cases where resistance has been documented. And so you see quite a broad range of antibiotics being affected in this case. So how is it at ILRI? Our focus as ILRI has really in different forms. And as I indicated earlier, we have a dedicated antimicrobial resistance, but we also have zoonotic team with different projects. And the focus is sampling in different forms, sampling at the wildlife, sampling within the community to be able to pick antibiotics, and to also pick samples from abattoirs, and then assess those samples for presence of pathogens, but also signals of antimicrobial resistance. Our focus has been basically on shotgun metagenomics, and I will be highlighting that in more details. Of course, there is those specific diagnostic tests like qPCR based. We have opportunity for whole genome sequencing, as well as metagenomics approaches. And how, therefore, do we focus in terms of AMR surveillance and One Health responses? These also require that we diversify in terms of protocols, in terms of even sampling techniques, and this has been presented by my colleague. I don't need to go into more details, but we still have challenges. There is technical limitations. Even the protocols that are available may not be robust enough. So the gaps need to be filled through coordination, collaboration with different partners. Within the African continent, institutions or countries are at different levels. So a platform for sharing protocols, opportunity for government commitment in terms of financial allocation for AMR and disease surveillance crucial. Of course, there is ethical issues, data sharing. Also need to come up with policies that allow robust data sharing without limitation. So we have actually two projects. One of them is under the GenPath Africa. The GenPath Africa is a consortium of collaboration funded by the Global Health Joint Undertaking, EDCTP3 Joint Undertaking, in also support of European Commission and Gates Foundation. So that brings a number of countries together, including South Africa, Mozambique, Kenya, and Europe, working together on a wastewater. Of course, wastewater epidemiology is just one of the packages. There are, a number of packages that is focused on. And for the Kenyan context, the wastewater surveillance is focused on two cities, that is Kisumu and Mombasa. We are focusing on grab sampling. We have dedicated sites along the sewer lines that have been marked. Kisumu, we have 18 sites. Mombasa, we have 12 sites. And these sites are along the sewer lines, and they are linked to specific communities. And so in terms of coordination, this has been done through the support of county health department, public health department in Mombasa, where we have been able to work with the leadership as well as identify, staff and field workers that we train and support. Beyond that, we are also focusing on feedback mechanism. Once data is generated on different pathogens, we organize for stakeholders engagement where we share the data and get feedback and support the public health response. The second project is the one that is between UN, UNEP, ILRI, and HERA, where we are focusing on the capacity building. And I think this is part of also that initiative. Of course, we are going to be focusing on beyond the capacity building. We are going to be focusing on transport hub sampling and data generation, I think my colleague has highlighted the process that we run samples through. So basically, the whole pipeline from sampling all the way to generation of metagenomic dataset analysis, the bioinformatic analysis, and communication of that data. And so this is just as a brief snapshot into the metagenomics dataset that we have generated so far. You can be able to see that over time there is a profile of different bacterial species, or a number of which are of public health interest. Of course, also bordering on zoonotic, and you can see there is a bit of a wavy movement around the abundance levels over time. The lower part of that, this graph, you can see the 1, 2, 3 is the number of weeks. So you can see over time, you can see as a pattern of a particular pathogen in terms of abundance up and down. And this is useful to inform public health in terms of response, or also identifying pathogens that are circulating before outbreaks. This is now part of the metagenomic dataset that is focusing on viral pathogens. Again, you can be able to see the pattern of the different pathogens in circulation and some form of fluctuation over time as well. And of course, this could be picked. You could zoom into different very specific pathogen from the metagenomics dataset, and you start to see now the patterns. And this pattern is what we hope can therefore support modeling, because we are hoping at some point in future we could be able to get retrospective clinical data set or concurrent clinical data set and model to be able to inform at what level should a response be initiated in terms of public health action. Now, I wanted to also highlight the fact that antimicrobial resistance has been shown to occur, and I think the opportunity for us to prepare should be emphasized by this, that you have the red coloring indicating when breakdown happens. The I think could be yellowish, greenish point showing deployment of specific antibiotics. And then you're able to see that over time, for example, if you look at salvarsan, this was first deployed in 1910. By 1925, the first report on breakdown of resistance. And you can see this has been demonstrated for a number of antibiotics. So this informs us that antibiotic resistance will occur. And therefore, we need a strategy and opportunity to be able to track, monitor, and come up with strategy. Again, this is just a profile of the data set we have pulled out from our metagenomics in terms of the abundance of different antibiotic resistance signals, and the specific drugs that are affected. So really the focus of WASE should actually be to inform public health. And it is important to think about the integration of WASE into the public health routine surveillance, but also letting the clinical data support modeling for integration of wastewater and environmental surveillance. I want to stop there and thank our team here, the donors and the government, the GenPath, and everybody who has supported this project. Thank you very much.
Thank you so much, Dr. Bernard and Jean-Pierre, for that good presentation. We'll go through a few questions that are in the chat. And since we've just finished with Dr. Bernard, I would like to start with you with a question from the chat. Considering that Jomo Kenyatta Airport in Kenya is a major hub in the region where many people change their airplanes, how do you take that into consideration with your data from aircraft arriving from there? Another example is Dubai, and the person is just wondering how these potential buyers of country of origin is addressed.
So should I go for it, or you are going to ask more before?
Yes, please, you can answer
Okay.
that one and then I'll, yes.
Yeah. The Jomo Kenyatta International Airport is already planned for sampling under the UNEP ILRI HERA project. And as the participant who has asked the question is very right in terms of it being an international transfer hub. And so it means that we have an opportunity to sample the airport and just look at the pathogens that are circulating to inform even nationally how to safeguard, but also to contribute to the global disease surveillance effort. So we hope that based on, and again, it is not possible, or it also in terms of ethical issues, we may not be able to tag, the flights in terms of the names, but generally it will be, through mapping of different destinations and different geographical, and routes, it is possible to largely associate the metagenomic status of pathogen just the areas of high risks, and then that could support, intercontinental collaboration and global surveillance effort to zoom into. So the device situation, it may not be possible really to really understand the specific individual or something like that, because that is part of data protection. But at least once we sample from an aircraft from a destination, it is possible then in a data sharing agreement that specific jurisdiction could be informed of, prevailing situation that could then help in the response that they take. But yes, we are focusing on JKA, and that data will help us just understand the pathogens that are circulating, including antimicrobial resistance. Thank you.
Thank you so much, Dr. Bernard, on that. The next question is for Jean-Pierre. And since this research covers a large population, is it funded by organizations like the UN or private groups? Also, is there any challenges you encountered with the community while doing your research? Jean-Pierre, please go ahead.
Yeah, thank you so much. So when we started, this was in the initiative was fully funded by the country, by the government. And we started supporting it with different projects that are funded by different grants. So some of it are the EDCTP3 for optimization of tools. So, and now we've started working with UNEP, but initially it was the country itself that was funding it. I can probably also talk to the question that Bernard has answered. So yeah, I understand the feeling of the colleague who asked this question about the bias that might be related with country of origin, since when you are collecting at a specific airport, so you will be having flights of different length and different origins. Some of them are connection flights, some of them are just stopovers. And there is an assumption when you are collecting from the aircraft, which might have its own bias. So the assumption of scheduling the emptying the tanks of the aircraft. Sometimes it's not at every airport where the aircraft lands, where where it's grounded. Sometimes the grounding is scheduled to the other airport, and this brings a challenge because it will be very difficult to really understand the results that you get from this aircraft wastewater if it represents which flights, because you are dealing with with waste that was deposited by more than just one flight. So that could be a challenge. But I think while keeping working through it, we can be able to deal with these issues and make it really more optimized. And one of the strategies which I think working together with ILRI, UNEP, and others would be to sample, doing an end-to-end sampling. Let's say if ILRI is sampling at Jomo Kenyatta and we are sampling at Kigali International Airport, at least there are several optimizations that can be done together by filtering the background noise, because at least we will have full background and full information about different flights. So at least with this approach, we can be able to solve those problems.
Thank you so much, Jean-Pierre, on that. We'll have one more question each, and then in the interest of time, please put your questions in the chat, and we will encourage the presenters to answer you in the chat. For Dr. Bernard, how did you do the antimicrobial resistant work? Was it direct detection or phenotypic or bacteriological work?
Yeah, thank you so much for the question. We basically use the metagenomics approach for our profiling of AMR. And this basically means what we do is pick samples, extract nucleic acid, subject them to library preparation, then next generation sequencing using the shotgun metagenomics approach. So what you end up with is basically the sequences, next generation sequences with largely the bacterial pathogens. So you get sequences of the bacterial pathogens. And so from the bacterial pathogens, then you profile the signals from those bacterial pathogens indicating genes of resistance to specific antibiotics. So we don't do culturing. We basically just map out the antimicrobial or antibiotic resistance through the bacterial sequences that we get from wastewater.
Thank you so much, Dr. Bernard, on that. The last question to Jean-Pierre: how do you manage to collect samples at the airport? What are the key pathologies you are looking for through metagenomics? How often do you collect samples at the airport? And what are your target sites? Thank you.
Yes, so if I understood well the question. So yeah, it's very complex to start collecting at the airport, especially when you're involved in aircrafts. So you have to deal with the airline companies to get the authorization, and also the management of the airport to get the authorization. So you can't start by collecting from every aircraft, because that's overwhelming, and also depends on how busy the airport is. So what we did was we established a system to help us to prioritize flights. So we were collecting from prioritized flights, and this was during COVID, and prioritization was in the context of COVID. So which means prioritization currently might be not the same as when we started, because now we are moving towards multi-pathogens. So we started by targeting SARS-CoV-2 as a model to optimize our system. But now we are moving to unbiased approach. So we are not, so currently we are focusing on not targeting any pathogen per se. We have a list of prioritized pathogens that we are having in the background of what we are developing, but we are, so like Bernard said, so we are working on shotgun metagenomics that really sequences without having in mind a pathogen. And then from there, we want to first establish genomic signature from the aircraft wastewater samples, and be able to also compare these signatures with those from well-identified communities, so community wastewater-based surveillance. And then from there, we can rework on our priority list, the list of priority pathogens, and see whether we carry with the pathogen agnostic or whether we keep the priority list. Because also something I saw Monica also was talking about the cost, because we also need to make a trade-off and see whether we keep going the pathogen agnostic way or whether we need to stick to an adjustable priority list of pathogens.
Thank you so much, Jean-Pierre and Dr. Bernard, for the great presentations. I was your moderator, Josephine Werimondubuti, a program associate here at UNEP, and I'll now hand over the program to Taro, my colleague, for the next session. Thank you. Over.
Thank you very much, Josephine, and thank you both presenters for the absolute great presentations and a very engaging discussion. I see the discussion is still ongoing in the chat, so please feel free, both presenters, to respond to questions. And if we are ahead of the time schedule in the end of the webinar, maybe we can still address some more questions also during the webinar. But next we will change the topic a bit. We will go to a stakeholder engagement session of this webinar. So let me introduce you the next presenter, who is Erik Lyma, who is a Tanzanian research scientist at the National Institute for Medical Research. And Erik leads wastewater and environmental genomic surveillance activities under EDCTP-funded ODIN Consortium, and where he is coordinating the field sampling, laboratory workflows, and data system across Tanzanian sites. So if you're there, Erik, feel free to share your presentation and your experiences about stakeholder engagement during implementation and designing of a WEST project.
Thank you, Taru. I'll be sharing my presentation.
Great, thank you.
I think you can see the full mode.
And if you can still place it as a presentation mode, now it seems
I've placed it as a presentation mode already.
Yes.
Is there as a presentation mode?
Not yet, but maybe it's just showing slowly to us.
Yeah. Mine, it's already in. I mean, on my side, it's already on the presentation mode.
Okay. It's not for you, Josephine? Is it only my screen or is it just being slow?
Also for me, it has not yet come in presentation mode. Yeah.
Okay.
Could you maybe try it?
And...
Okay, now something is happening. Let's see.
Okay. What about there? Otherwise, I can try another method. Let me.
Still shows like a normal PowerPoint slide, so let's see if we can.
Let me share the window.
Yes, thank you.
Probably entire screen. Share there, then I bring it here. And then what about there?
Now it's yes, in the presentation mode. Thank you.
Oh, perfect. My name is Eric Limo from the Tanzania Institute of Medical Research, and I'll be sharing lessons we've learned in the ODIN project about engaging stakeholders for wastewater environmental surveillance, or here we are calling it WES. So ODIN project is a project which has been funded by the EU EDCP, and we are currently doing environmental surveillance and wastewater analysis. And before we start our project, we engaged stakeholders, and our journey is much more about much more than collecting samples. As you heard, that I'm supervising sample collection and the analysis of the National Institute of Medical Research in collaboration with the National Public Health Laboratory. So our aim is about building trust and collaboration in sustainable systems. So our project is one of the first ones to do this kind of a project in a large scale and analyzing, priority pathogens. So we are also trying to see how this work can be carried forward after the project ends. So in summary, I'll take you through the background, our approach, and ODIN achievement and key achievement, challenges, and finally the lessons we think are relevant not to Africa but also globally. So starting with the background, why WES? Why wastewater environmental surveillance? This is because traditional clinical surveillance tells us a lot, but it has limitations. So it often misses asymptomatic cases, and it can be slow, and also it can be costly. So wastewater environmental surveillance is emerging as a powerful tool that complements clinical data, and it also gives us a cost-effective and comprehensive view of community health and allows for earlier detection of outbreaks. So this is in the context in which ODIN was established. So currently, we've seen a lot of waste, especially targeting the SARS-CoV-2, since we are just coming out of the large, pandemic. And before, it was just other disease-causing organisms and probably drugs and pharmaceuticals in the wastewater. But now I think SARS-CoV-2 has taken a center stage in the wastewater surveillance to act as a public warning, and also currently is the mpox. It is now coming up as one of the components for wastewater surveillance. So overview of the project, as I said before, it's a EDTP funded project in Tanzania, Burkina Faso, and DRC. We also have our northern partner from Finland, Belgium, Sweden, and the UK as our mentors. And also at its core, ODIN aims to strengthen genomic and the bioinformatics capacity across these three African countries. So a central feature for the project has been stakeholder engagement. This is one of the activities we did very early in the before starting any scientific work or any surveillance work. It's because the environmental surveillance systems succeed when people responsible for implementing and sustaining them are fully involved from the start. So from there, you can get on what to surveil in the environment. So to start with, stakeholder mapping and engagement. So you have to do mapping and to see who you can engage. So at the beginning, and also as you are going along, conducting a wastewater environment surveillance project or a program. So for us, we used the TGHN stakeholder mapping tool was used. And this is, THN is one of, at the Oxford University is one of our of the consortium members. So Odin is I'll show you in the first slide. Odin is a consortium, and THN from the Oxford University are one of the consortium members. So using the tool, so we identified as policymakers, academia, NGOs, the authorities as one of our stakeholders. And when we engaged them before we start the project, there was high participation across the 3 countries. So each country did its own workshop. And as we can see here, a participant by country. In Tanzania, we had 29 stakeholders. In Burkina Faso, there are 40, and in DRC, there were 26. So we brought together a wide range of actors, as I told you before, and this number you are seeing here in the slides. Our method included a national workshop, interactive surveys like Mentimeter. Also, we did a field visit to wastewater sites, and also we had hands-on tools such as the Odin Lab Handbook, which has been published. So the handbook details on how we are doing the environmental surveillance. This is a more technical document that we've been able to come up with. So the principle is not just to inform stakeholders, you have to involve them. So we started with few stakeholders before we went to the community. So as I said before, we had hybrid workshops, surveys, field visit, and discussions, discussing potential surveillance, potential challenges and benefits for this for wastewater environmental surveillance. So as I told before, lastly we had the handbook and of course the training and dashboards. So as I told you before, we had 29, 40, and 26 participants from across the 3 countries, and you can see we had academic researchers and professional associations. We had policymakers. These are the ministries and also the National Public Health Laboratory. We also had policy networks and international and regional organization. So this includes countries office such as the World Health Organization and the UNICEF, and also from the Water Aid. We had these participants in our workshop when we are launching the environmental surveillance. We also had several non-governmental and civil society organizations. So at the end of the stakeholder meeting, we came up with a publication at the International Journal of Hygiene and Environmental Science. The publication is Strengthening Pathogen and Antimicrobial Resistance Surveillance through environmental monitoring in Sub-Saharan Africa. So these were the stakeholders' perspective. So these were results. These were perspectives from the stakeholders. And from them, we wanted to see, which, areas of public interest that we should look when we are doing the environmental surveillance. So we were able to get, a priority of pathogens that were of public importance. And for waterborne diseases, stakeholders prioritized polio, salmonella, typhi, and cholera. But since polio is already among, polio has its own program in every country. So polio has been a program, and there's a polio program which is doing wastewater surveillance. So we had to jump to salmonella, typhi, vibrio cholera, and other other targets. So beyond pathogens, we identified gaps. Beyond pathogens that are of public health importance, we also identified gaps. this included limited labs, weak coordination, and policy gaps. This is what we got from the stakeholders. But important, stakeholders also saw the benefits and the early detection and the better preparedness and stronger resource allocation and the awareness raising that that can unlock new funding and also opportunity for further programs. So I mean, during the workshop, so we had organized the workshop in 4 thematic areas. This included clinical surveillance as currently implemented, ongoing environmental surveillance practices. From there, we were able to realize that polio is a very strong program that has been running. Has been running for many years, and also it had a lot of success. In curbing the outbreak of polio in several areas. So also we are able to identify key challenges facing both clinical and the environmental systems, and also opportunities. So during this stakeholder engagement, we saw the opportunities of potential for wastewater and environmental surveillance. So when we were engaging the stakeholders, we were able to see what also has been happening in the developed countries. So we look globally. There are lessons that we could adopt in our project. This is what we got from the stakeholders. What we saw, example in the US, diverse stakeholders networks and dashboards helps track the COVID-19 real time. So in developing countries, they were able to quickly start the COVID-19 surveillance, the SARS-CoV-2 surveillance through the wastewater. And in Europe, they were able to harmonize protocols, strong community engagement, and have been central, especially in Europe. And ODIM was our project was able to adopt these ideas, but we had to change them for our low resource and non-sewer context. As you know, most of our cities in the African countries, they are not connected in this sewer network. So we had to, through the stakeholder engagement, we got some tips on how we can go around those hardships. So what we saw from the stakeholders meetings, that challenges are not they are not opportunities. I mean, challenges are not barriers, but they are opportunity to innovate when you are doing this kind of when you're doing work with stakeholders. So as I told you before, we saw there is a weak coordination across sectors. Also, there is poor infrastructures, but we are able to find solutions for some of these challenges, especially for non-sewered system and poor infrastructures. So what the solution was to do a stepwise approaching, include desampling from open drains, rivers, and public latrines. Some of the stakeholders they informed us that some of the communities, wastewater is being let loose and go into the environment. In some of the streams, it also goes to the urban rivers. And we are able to identify some of the wastewater is going directly into the ocean. So when you're doing stakeholder engagement, there are best practices, and this, stakeholders will come in many groups. So there will be these professionals which we need to engage them before you start with, and also the community is one of the great stakeholders. So you should also have structure them on how to engage. So you have to plan early and systematic on how to engage its stakeholders, including the government side, the NGOs, and also at the end the community. Also in that you have to tailor your communication and the message according to the stakeholder groups. I'll show you the examples of that. Also, you have to maintain transparency and accountability. So you have to be very transparent especially when you are giving the results. Also ensure that there is a feedback loop. So when you're doing environmental surveillance, you'll be going there maybe weekly or monthly or every day. So the stakeholders, especially the community, they will see you. So you have to give them some some of the results that you are still getting. We know some of these programs or projects, they are running for 1 or 2, 3 years. So you have to have a kind of mechanism of feedback to the community, and that will promote trust and ownership of such programs. So, and one thing that we can do that is sustainable waste through One Health collaborations. So to involve all parties of during the environment surveillance. So I'll take you back on how to framework the stakeholder mapping. So one of the key, there are two main things that you have to consider: is the primary interest and motivation, and also potential concerns. This potential concerns is also applied to either public officials, water sanitations authorities, or the community itself. So I will touch 4 of these. To start with is the public health officials as a stakeholder group, and this will include the epidemiologists, and mostly from the Ministry of Health or Ministry of Livestock or Ministry of Water. So it depends on your setting. And these are the primary interest that you need to engage them, is that these will serve as early warning authorities. They also need to understand the true disease burden, especially if you are targeting disease of public importance. And also potential concerns is how they can interpret the data. And also resource burden, if there is something came up from the environmental surveillance, you need to also operationalize the wastewater data. And to go with to the fourth row on this slide, the community and civil society. So these will mostly be the community leaders, NGOs, of course, and the medias, because these are the ones that will give you take far your results. So it will involve transparency, as I said before. You have to have the information accessible to them, and also you have to empower once you see some of the interesting results from your surveillance. But you have to be careful when you are dealing with the community, because this can raise stigma, especially when you are doing sewage surveillance, because you can start to see some funny results there. For us, we also did clean water surveillance. So there is even more stigma in that if you find some contaminated clean water in some of the society. So the data can be misinterpreted. So getting pathogens of public interest in the clean water does not really direct it to the outbreak in real time. So you have to be careful when you are decimating the results. So these are the 4 stakeholders that I've touched with. These are, so I want to stress in West, the public and community are the most at the top of the priority. But this is when you are disseminating your results. But when you are conducting West, the water utilities. In our settings, the water utilities they deal with the clean water and also with the wastewater treatment. So these are one of the 2 top stakeholders in West. And at the beginning, and you have to involve the public health officials. But at the end, and at the beginning, you have to involve the policymakers. So as you are doing West, you have to involve policymakers. At the beginning, and also at the end, when you want to have a data-driven leadership and cost effectiveness in curb some of the diseases of potential outbreaks. So how can you measure success when you do stakeholders engagement? So it can be indicators such as participation, especially for the public health officials and from the community, and also if you are doing West, you have to measure your success by looking at the consensus. So this is between the public officials and the policymakers on if you are getting some interesting results in West, you have to see how do you reach consensus and how do you adopt, policy, especially if you are doing surveillance in clean and the wastewater. And also, as I told you before, there is a challenge in African countries. So you'll be sampling in open waters, in running waters, in open sewers, in trenches. So once you get some funny results, you have to see how you can change the policy in a certain area. So for us, Odin, some of the outcomes are publication, as I told you before. And also, we are going to have more publications. We will publish the stakeholders' perspective. We will also publish the handbook, which involves sample collection, analysis, and the results reporting. And also, we have been able to train. So because WES is quite a new program in the 3 countries, so we have been able to train labs and health public labs to do WES surveillance. So also in that way, you can have a short, mid-term, and long-term, impact. So one of the long-term impact is to integrate One Health surveillance systems. So to go quickly, effective WES, it will also involve ethical and governance issues. So these are 4 in our perspective. We are saying that you have to respect privacy when you get when you are doing WES. You also have to avoid stigma when you have results and you want to disseminate them, because stigmatization is a risk, because you can be doing WES in a community or in a city in different areas. So you have different results. So you have to see how you can minimize stigma. Also, you can avoid blaming any community and use a neutral and careful language. Because as I told you before, one of the key thing when you are doing WES and there is no connected sewer networks, so you have to do environmental surveillance in soil and the running water and the streams across urban areas. So if you get maybe so much column forming units, or you get cholera, or you get what, so you have to be careful on how to disseminate your result. You also have to develop a legal framework. Policies must be defined of ownership. You have to define data access rules. Now there's a social media thing. It has a lot of impact. So once you have the result, you have to really be careful on how to deal with the data. Also, you have to integrate into existing laws when you have results from the WES. And also, we can always cope what is happening in the US and in the EU, where they really do have a very strong WES programs. Also, you have to ensure transparency, enable cross-sector data flow, and balance openness with security. So, effective WES framework, we are proposing to have 4 pillars. That's regional collaboration, standardization and common tools to enhance data comparability and coordination when you are doing WES across several countries or several places within one countries. You have to have a national integration into health plans once you have WES results. Also, you have to have a shared investment. That's collaborative funding, builds ownership, resilience in health infrastructure. So you have to have common programs across certain areas. So what we have achieved, I will go quickly. This involves One Health gaps.
Eric, just a quick note, if you can wrap up with the one slide, so we have still some time for questions to pose. Thank you.
Thank you. So what we have achieved, I'll go now quickly, is the One Health gaps. We've saw that through the stakeholder engagement. We've also that we have also seen that we can leverage the existing polio surveillance that I told you before. So for us, Holding has been recognized as a pioneering WES project. This is through the engagement of stakeholders and have a very structured, program. And we have been extended to do the mpox surveillance in hubs. So we have started airport sampling, like one of our colleagues said. So currently, in addition to the environmental surveillance in several places, I mean, in the established area, we're also doing wastewater surveillance at regional hubs. So in conclusion, we've seen that how wastewater environmental surveillance can move from being just a technical tool to becoming a system built on trust, ownership, and collaboration. So the Odin project has shown that that with the right engagement, even the toughest challenge, whether unsecured or weak coordination or limited infrastructures, can be transformed into opportunities for innovations. This is especially for developing countries. So I'd like to leave you with one thought that waste is not only about detecting pathogens, it's about protecting people and building resilient health systems together. So at the end, what you have to consider mainly is which stakeholders are most critical and which are the hardest to engage before you do your waste. So thank you. This will mark my end of the presentation. Over to you, Tab.
Excellent. Maybe I can jump in now. I'm Kate Medlicott from the World Health Organization, working on our wastewater surveillance program. I have just a couple of questions too, and I think, you really set out a very comprehensive stakeholder engagement approach. I can imagine that's really a lot of work and quite time consuming to do all that. And you finished with almost exactly the question I was going to ask first, like, who are the most important stakeholders in all of this, and which ones were the most difficult to engage? What are the lessons there?
Thank you, Kate. I think the most important ones are the public health officials. These are the ministries, because they develop policies, and the policymakers are the important one, but they are the top ones, the legislative ones, and the top and the top guys, which you can't really meet them. So the most ones are the public health officials and the water utilities, because these are the ones that they deal with clean water and also wastewater treatment. And lastly is the community. So for us, we were able even to sample the markets and the environment and the streets. So these are the people who are very interested on the results. And which ones are difficult, I think, are the policymakers, because these are top level officials. So getting them in one place and deliver a message is one of the difficult tasks. Yeah.
Yeah, that makes a lot of sense, because as you say, it's not just about the detection of pathogens, but actually getting to that decision and an action related to it. And just sort of looking now at some of the sort of techniques you talked about, workshops, surveys, field visits, some of the planning, communications, ensuring transparency. What of those sort of techniques do you feel was the most effective, that was really giving you the most return on the effort you're putting in, and how were you able to distribute that work across the various stakeholders?
Yeah, so I think the most important ones is the survey. So the survey, when you involve these, especially public health officials, NGOs, and some of the leaders who are, especially the responsiveness team. I don't know if that's common across countries, the one who are dealing with outbreaks. So these are the most important ones, and they're the ones that need to understand your methodology. And also these are the first ones to get your results once you do this surveillance. Yeah, so so that's kind of a framework. So those are some of the stakeholders that you need to engage very early. And to the water authorities, they do have their own labs too. So you also have your analysis coming up. So there might be some clashes and some disagreements on which results are. For us, we are able to do very top science, because as I said before, we have this it's a consortium. So we have even newer techniques from Europe that we are adopting now. So sometimes it's difficult to change, to communicate the message and for them to take it for policy change. So yeah, so that's our experience. Yeah,
that's right. I think what we see quite consistently is that all of these stakeholders are at a different stage in their understanding of what is the value of wastewater surveillance for them, what would it take to integrate it into their work, so that it's a very nuanced kind of engagement we need to have with each of those groups to demonstrate the potential and added value. I have so many more things I would love to ask you, Eric, but we're at the end of the time we have available, so I'm going to pass back to you, Tara, for the closing. Thank you.
Thank you very much, Eric, for your presentation, and Kate for posing the questions. And yes, of course, if you still have any questions for either any of the presenters, please place them to the chat, and hopefully the presenters can still keep going answering those. As said, we're coming to the end of the webinar, and I would like to thank you all very much, all of our speakers, and of course you participants, for today's engaging session. And yes, we have heard valuable insights on collaboration across the sectors, and also essential for advancing WES in Africa. And we hope that this inspired you and all the upcoming webinars also, and gave you some new ideas and connections and opportunities for collaborations. And with that, we would like to encourage everyone to take the first step to share this webinar with a colleague, join working groups in your country or within international initiatives, or commit to change one change in your topic area that enhances collaboration across these sectors. And of course, it would be great if you would share your planned next steps in the chat to inspire others. So, because that's what we want this platform to be, to inspire everyone who are attending. So on behalf of the organizers, I would like to thank you once again for your time and contribution. And as said in the beginning of the webinar, we have made some post-webinar survey that will be soon. There will be soon a link and a QR code in the chat. And we really wish you all to answer this survey, as there will be also questions about which topics you would like to see in our next webinars, as we will have altogether 8 webinars, so 6 more to come. So there is really a big chance for you to also further affect the next topics. And just giving you a few minutes to see that everyone are able to access the survey, the link, and start filling it. And I think in the meantime, my colleague Josephine would like to take a picture of our webinar participants. So if everyone could turn on the camera, let's hope that the connection will last that. So for a second, so then we can take one group picture to remind about the successful webinar. So that would be great. Thank you. And if you are having any troubles going to the survey, then let us know and we will try to fix the technical problems. Great to see all of your faces also. And yes, there were a couple of questions also in the chat about the recordings and then the presentation. So the recordings will be available. We are planning to have a training online platform or a knowledge tool within this initiative. So the webinar recordings will be embedded there and also available in our webpage. And also we will be sharing the presentations for you later on. And yes, we look forward to continuing this dialogue and seeing you all in our upcoming sessions. And with that, I wish you all a wonderful rest of the day or evening, wherever you are. And thank you all.
Thank you so much.
Thank
Thank
you.
Yeah. Thank you.
Bye.
Bye.
Thank you.
Thank you.
Thank you.